Okay, as I said, today we want to look at bacteriology. Um, Previously, we had said that bacteriology is the study of bacteria, which is a sub branch of microbiology. Of course, the people who studies bacteriology are called bacteriologists. Um, uh, According to the history, uh, bacteriology started way back in around 19th century, where several discoveries were made. But one important discovery that contributed a lot concerning bacteriology is this scientist by the name, Anton Lowenstock. So this man, Anton Lowenstock, he discovered bacteria in 1676, where he used a single lens microscope, which he had discovered to see these microorganisms, and he named them molecules. So molecules, according to Adams van Wenshoek, they are small animals which cannot be seen by naked eyes. Later, the likes of Louis Pasteur, who was a chemist, was able to recognize the living organisms which were able to cause what you call fermentation of wine and souring of milk. Thereafter, we also have people like Robert Koch, who was a German physician. He was able to grow bacteria in culture and they could be able to purify and subculture it. Doing that in what we call the famous Robert Koch postulates. He was able to, convince people that indeed uh, this bacteria exist. In around 1884, there was this Hans Christian Gram who discovered that some bacteria could stain blue with what he called crystal violet, and he was able to confirm that some could not be able to, stain, and therefore they were also given a different name, as we'll see later. So, according to classification, after some time, it was realized that some of the factors that determine classification were what we call the RNA sequences, the shape by chemistry, and some of the characteristics of the external membrane, e.g., cell wall and flagella, were among the characteristics that were used to classify bacteria. So, according to taxonomy, the bacteria were ranked from what we call domain, being the kingdom, then domain, the phylum, domain, kingdom, phylum, class, order, family, genus, and species. You realize that for the viruses, the way I told you, we only classify them from the family level, genus, then species. But like bacteria, they were well classified because they had more of a complete characteristics of living things. So they come way back to what we call domain. Currently, classification of life are in three domains. We have the eukaryotes, prokaryotes, which are the bacteria, and the archaeas. So when you talk about the domain, we know that bacteria are under what we call prokaryotes. Archaeas are also simply single-celled microorganisms, also sometimes called prokaryotes, and where they have no cell nuclear or any other membrane-bound organelles in their cells. So that can also be a way of how to differentiate the bacteria who have cell organelles, a complete cell membrane, or as opposed to archaeas, which are single-celled but they do not have nucleus, neither do they have other uh, membrane-bound organelles. They were initially classified as bacteria, but receiving the name Archaebacteria in the kingdom Monera. Thereafter, we have the eukaryotes, which is include the organism like all animals, plants, fungi, and protists. Fungi are big enough to be under the group of eukaryotes because they are multicellular. But bacteria show an extreme degree of diversity and by the number make up a large majority of the known species. No? And therefore, that's why for now we cannot claim to be classifying like all the bacteria per se because many are still not, classified as per now. So bacterial classification identification, one of the key things that was used to classify bacteria is the shape. And this is the simplest way to classify bacteria. There are several shapes that are associated with classification of bacteria, which includes some bacteria are oval, others are rods, others are spiral, and others sphere in terms of shape. Um, so, as such, shape is not necessarily an exclusive characteristic, meaning you can't say that, you know, Wamalu and Masinde look the same or they're male, therefore they're the same. That shape alone cannot be able to classify or characterize bacteria because and then it only gives it in a broad based classification. Again, same to size. Size can also be used to classify these bacteria significantly into species, um, but many species tend to form groups of individual cells which can vary greatly in number. So the presence of external structure, apart from shape and size, external structure can also be used, to classify bacteria. Like for example, we have sometimes what we call tentacle-like constructions called flagella. This flagella can help distinguish bacteria species because some have, others do not have. Another way is what we call staining technique, what we call gram stain. So gram stain is another key tool in bacterial classification. In fact, as we speak, is one of the fundamental tool that is used to assist medics in terms of classifying bacteria. You realize that this gram stain is a test which was discovered by a scientist by the name Danish bacteriologist named Hans Christian Graham. Hans Christian Graham in 1882. This scientist, Hans Christian Graham, is the one who discovered this gram stain technique. And therefore, because he was the one, the name gram stain was therefore used. And that was way back in 1882. So this gram stain quantifies bacteria according to the thickness of their exterior membrane and therefore can classify bacteria into two major categories, what we call gram positive and gram negative. Yeah, so when we talk about gram negative bacteria, they have a very thin membrane, and the gram positive bacteria have a thicker membrane. That's hence the word gram staining is used. So metabolism is another aid that can help us to classify bacteria, where others normally carry out the metabolism under what we call oxygen environment. Hence, we call them aerobic bacteria or aerobes. And then those which carry out metabolism in the absence of oxygen or in the presence of CO2. Such are called anaerobic or anaerobe bacteria. And then we have the biochemical distinctions. Some bacteria can ferment, some carbohydrate, others not. That can also be used to classify bacteria in different categories. But more importantly, of recent, Best way, sure way of classifying bacteria is using what we call molecular analysis. This is a recent advancement where a technique that's made possible to bacteriologists to differentiate bacteria according to the differences in RNA as well as specific gene sequences. So, based on this now, therefore, further analysis of the total amount of individual RNA and DNA proteins and the ratios are also a way that can be used to classify. However, using some or all these techniques and observable characteristics, then bacteriologists are able to classify bacteria according to species. So those seven possible ways can be used to classify bacteria, right from the domain up to the species level. So in terms of shape, is commonly used even in the lab to differentiate bacteria. Those bacteria which we call cocci are what in the terms of morphology is the same as shape. The cocci, when you talk about cocci, these are round shaped, bacteria. When you talk about coccobacilli, these are ovoid. When you talk of bacilli, these are rod shaped bacteria. Fus- Fusiform are pointed end, curved or spiral shaped bacteria. So that's the common shapes we normally have. You will keep on now seeing them in what we call patient requests. Lab people keep on describing that. Maybe I've seen cocci, this bacilli, or even rod. The other name for the bacilli is rod. So we also look at like the cellular arrangement as a way of classifying bacteria. Most clinically important bacteria species range in size from 0.25 to 1 micrometer in width and 1 to 3 micrometer in length. Uh, when you talk about the bacterial cell composition, here the composition are divided into cells that make the cell envelope and its appendages, and those that are associated with what you call the cell's interior. For example, some bacteria have what you call an outer membrane, and this outer membrane is only found in gram-negative bacteria. And we are talking about now the ones with what we call cell envelope. So they have an outer membrane, and this one is only found in gram-positive negative bacteria. And then we have cell wall that is made of peptidoglycan or murine layer. And then we have periplasm. Periplasm only found in gram-negative bacteria. Then the cytoplasmic membrane. And what is the function? It gives the bacteria cell its shape, and it gives it the strength to withstand changes in the outside environment, environmental osmotic pressure that can cause cell lysis. It protects against mechanical disruption of the cell and serves as a barrier to larger substances. And it contains several antimicrobial agents and confers virulence. So I am taking time to let you know this because as you'll be graduating and going the field, you will deal with most of the microorganisms that cause disease. And therefore, when you hear a gram negative bacteria, or a gram positive, you must know where does this microorganism draw the source to to be pathogenic, or to be virulent, or to cause disease. So, in a nutshell, or in summary, this is what we say. For gram positive cell walls or gram positive bacteria cell walls, number one, it has 90% of peptidoglycan, which is thick and it is several layers together. Number two, it contains in the cell wall contains gram in the cell wall of the gram positive bacteria. It contains what we call teichoic acid. Teichoic acid, which is linked to the peptidoglycan layer. Number two, it has what we call lipoteichoic acid. This is simply lipid plus teichoic acid, which is used to span or to rotate the peptidoglycan layer, and is linked to what we call the plasma membrane. So that is the composition of a gram-positive cell wall bacteria. Then a gram-negative bacteria cell wall, number one, it has what we call thinner peptidoglycan layer, which is simply 10% peptidoglycan. But this 10% thickness of peptidoglycan in this gram-negative, it is attached to another outer membrane via what we call lipoproteins, and this outer membrane is 90%. Remember, in gram positive, it does not have outer membrane. It only have peptidoglycan, which is 90%. In gram negative, peptidoglycan is only 10% thin, but it has an extra layer, an envelope, which is 90%. And again, gram negative do not have teichoic acid in its cell wall. So this outer membrane, we are talking about 90% in the negative cell wall bacteria. It has it comprises of what we call lipopolysaccharides, lipoproteins, and phospholipids. That is the composition of the cell wall of a gram-negative bacteria. Of course, it also has a periplasm, which is a space between the outer membrane and the plasma membrane. So after saying that, uh, how do we identify bacteria in the lab? Bacteria are routinely identified using the following methods. We can use shape to identify bacteria in the lab. We can use biochemical tests. I talked about fermentation. There are some bacteria which ferment carbohydrate, like for example, let's say sucrose, others ferment lactose, like that. And then we can also use serotyping, where we use the principle of antibody antigen identification. That one you've covered in immunology. We can also do culture. We can grow bacteria in the lab so that, like for example, when a fetus is still in the body, we may not be able to, like for example, we have mothers, 10 of them here, and we screen through ultrasound or CT scan. And then we pick the pictures of those fetus inside the homes of the mothers and put all of them here. When I ask you to identify them, you cannot identify them. You cannot associate with any identification. But when you let these fetus grow, mature, be delivered, they grow up, you could be able to say, this is Wamalwa's son, this is Precious' daughter, this is Masinde's nephew. Why? Because of the physical features that come up. And that one is under what we call culture. We also have molecular technique which identify these bacteria up to the level of the genome, the genetic material, which is more a surest way of identification. Um, so that is how it is. And then we have when you talk about growth characteristics or the culture, after the culture has been made, a primary distinguishing characteristic is whether an organism grows. There are those which grows aerobically, that means under the presence of oxygen. Others grow anaerobically in the presence of CO2 or the absence of oxygen. So it means you must avail or supply or provide that kind of environment for that particular microorganism to grow. For example, if gonorrhea bacteria is anaerobic bacteria, when I provide anaerobic condition, they will not grow. That's why you use this condition to, as a method of differentiating or identifying bacteria. Others that are facultative, that is, they can either grow in the presence or absence of oxygen. You see, with or without. Others are what you call microaerobic. That is, they grow in the presence of less than atmospheric partial pressure of oxygen. Oxygen kidogo. Okay. So apart from that condition, we also have what we call the temperature. Many bacteria grow in different temperatures. So temperature can be used to as a characteristic to differentiate bacteria in terms of growth. We have pH, nutrients required. Others maybe need carbohydrate. Others only need blood. You know, like for example, those, let's say when if you are culturing, let's say malaria parasite, without supplying red blood cells with the hemoglobin for them to grow inside the red blood cells, they will not grow. So these are some of the conditions you must understand under which condition does this bacteria grow, so that you provide the same condition in the lab for them to grow before you identify them. And some are even resistant to antibiotic. Uh, this might not really be of interest, but it's just good to mention. When you talk of culture media, these are substance or foods that we use in the lab to grow bacteria. Some of them we call them selective media. Why? Because they contain some substance that inhibit growth of certain microorganisms and allow others to grow. That's why this is called selective. Like for example, if I take a certain antibiotic and mix with a media, maybe I'm targeting gram-negative bacteria not to grow, but I only want gram-negative to grow, or vice versa. If I only want gram-positive, I put an antibiotic in that media that inhibit any growth of gram-negative, so that I only remain with one. So anything that is used to suppress the growth of one group and allow another group to grow, it makes that media to be called selective. For example, the high salt concentration in mannitol salt agar, it inhibits the growth of most microorganisms except Staphylococcus species. you know. And then there is what we call differential media. This one allows multiple bacteria species to grow, but their growth patterns differ visually. Yeah. For example, you can have a media that has different components. Let's say if salmonella grows, it can be pink. If staphylococcus grows, it can be yellow, and another one can be pale, or colorless. So that's why it is differentiating. It has different components that can give different patterns, hence allowing you as a scientist to differentiate. For example, when you have what we call blood agar, is a commonly used differential media which contains 5 to 10% sheep or horse blood. This blood is a requirement for the growth of bacteria like Streptococcus species that grow, and by so doing, it hemolyzes the blood. And when the hemolysis is done, we are able to differentiate because other bacteria are not able to do that. About serology, I talked about it, is the check for the presence of level specific antibodies in the body or in the blood produced against a specific bacteria, and several of them are normally used, you know, to differentiate some of the bacteria. Molecular technique analyze biological markers in the genome and the proteomic, um, just to know if um, a certain bacteria genetically is related to another family or not. So this is the, as I say, the finest way. So I have just summarily given that kind of background so that we understand when we talk about gram negative bacteria, we know what it means. Gram positive, we know what it means. We talk of cocci, we know what it means. When you talk about bacilli or rod shaped, we know what it means.